Right. Good evening, people. Good evening. Can you hear me clearly? Can you hear me clearly? Yes, Ned. You can hear me clearly, right? Good evening. Good evening. So, what's news these days, people? And did you go? Today is holiday, ne? Poe day holiday, right? Right. Hello, hello, Mr. Rimas. Hello, people. Today is a big day. We're going to discuss a couple of the previous theory paths. I thought of discussing the plant life cycle flowering plants, Anthophyta, and then we have to discuss people fungi a little bit, and then cell cycle, right? So we'll start, Ned. We'll start the discussion. Right. People, plant life cycle. We'll start with plant life cycle, right? Plant life cycle part, right? Plant life cycles. So, plant life cycle Anthophyta. So, in previous discussions, we went through the Bryophyta, Pterophyta, and then Psychodophyta, right? Selaginella also we discuss. So today's discussion starts with Anthophyta life cycle. But I saw last time, right, still students are weaker in this plant life cycle. I think they didn't attend for this discussion. So it's better, ne? Better you continue, right? So go to my plan, and that is better. So people, life cycle of flowering plants. Life cycle of flowering plants. Anthophyta. Structure and function associated with sexual reproduction of flowering plants. Sexual reproduction of flowering plants. Right. People, flowering plants, Anthophyta. Anthophytes. What do you know about anthophytes? Flowering plants. What's the dominant stage? Flower is the dominant stage. Sorry, the sporophyte is the dominant stage. Sorry. Sporophyte is the dominant stage. And then we can see people, we learn about the formation of the flower also. Formation of flower, right? Formation of flower. We learn flower is a special reproductive structure, but flower is a structure with microsporophylls. It's a collection of microsporophylls and megasporophylls. Cluster of microsporophylls and megasporophylls, right? So let's pay attention to that. The flower formation of flowers. Right, look at this. Look at this. Flower formation. So this is the flower. You can name these parts. What is this part, people? Can someone tell me what that part is? You learn, huh? This is not the first time you're learning it. What do you call that? Receptacle, the receptacle. And what are these two people? Right? What are those two? Come on, be live in the chat, right? So, what are these people? Rimas, do you know what they are? Amna, Abdullah. So, what is this one? What do you call it? Come on, calyx, right? Calyx, calyx. And then we learn people. The next one is the what is this? Corolla. This is corolla. 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 And then we have this. What is this? Come on. Come on. This is the male part of the flower. What is it? Male part of the flower. Right? Yes. What's it? Come on. Please send. I have opened this main chat. Have I open main chat for you so you can chat with me. I think our main chat is open. Is it logged? Everyone, possible. Not logged. Androsium, very good. Androsium. Come to the main chat. Stamen. Okay. Stamen or androsium. What's the difference between the stamen and the androsium? One is a stamen. Everything together we can call as the androsium. And this one is the gynosium. This is the gynosium. So we talked about this formation. We learn people androsium formed by the microsporophyll. If you think of Selaginella, this is the microsporophyll. Microsporophyll of Selaginella. So we believe that the anther has evolved from that. And you learn people the megasporangium. You learn the megasporangium of Selaginella. This is the megasporophyll. Megasporophyll. That's megasporophyll. So this is microsporophyll. And what is this? Microsporangium. So microsporangium has turned into the anther. This is megasporangium had turned into the gynosium, right, people? This is the gynosium. So gynosium has formed from that. Okay. So in other words, we learn people these are modified reproductive leaves are forming the flower. So flower is a reproductive structure. Flower is a reproductive structure. Right? Flower is a reproductive structure. So we learn this flowering plant life cycle. We learn life cycle of the flowering plants. Right? You learn this. And we labeled this diagram also. We label this diagram. So you know now this is a modified microsporophyll and megasporophyll collection. Modified microsporophyll and megasporophyll connection. Right? Right. Oops. Oops. Wait. Oh, something wrong. All right. You see some lines, something wrong. Okay. Doesn't matter. We'll continue like that. Okay. Now look at this. Look at this. This one is the sporophyte. What is this? Sporophyte. Sporophyte. This is sporophyte. And then here we can see the flower. We can see the flower. And then you can see people these parts of the flower. This is flower. Flower paths. What are flower paths? Number 3, stamen. Stamen. What's number 4? Gynoecium. Gynoecium. What is gynoecium? Gynoecium is the female part, right? Gynoecium. Then we come to the stamen. If you look at the stamen, parts of the stamen, this is anther. Anther, ne? Anther. What is this number 8, people? Can someone tell me what number 8 is? Filament, ne dalamai? That is filament. Filament. Number 5 is, I'll take stamen. Number 5, stamen. I would like to see that you follow your books, right? If you can follow your books, better, right? If you can follow your life cycle. And then people, number 9. Number 9, let me take as this one. Can anyone tell what that is? It's called antelope. Antelope. Number 10, my dear. What's number 10? Shall we take as microspore mother cells? Microspore mother cells. Umma cells. Mother cells. Mother cells. You call umma? Umma for mother. So this is microspore umma cells. What is the ploid level of these cells, people? What is the ploid level of the cells? 2N. 2N cells. So what happened to those cells? They undergo meiosis. They undergo meiosis. Result of meiosis, what's that? What is the result of meiosis? It forms a microspores. Microspore. Microspore. What is the ploid level? Haploid. It is a haploid cell, microspore. Right? It forms the haploid microspore. It forms haploid microspore. Right? So this microspore becomes the pollen. It becomes the pollen. Right? So this is number 12 and 13. We say that the pollen wall, right? Pollen wall, the wall of pollen. It has names called exine and intine. You don't need that. But if someone can tell what number 14 is, he or she is going to make my day. What's number 14? Come on, who's going to tell me? Today attendance is good, ne? Somehow people like theory, ne? So what is number 14? Who's going to tell? Who's going to tell that? Number 14. No one. No, you have to chat with the, come on. You have to chat with the main chat line. Generative nucleus slow. Okay, polar nuclei. No. I'll agree with that. Which one? This is generative. Generative nucleus. What is the other one called, people? Tube nucleus. The other one is called as tube nucleus. Tube nucleus. Tube nucleus. Generative nucleus and the tube nucleus. So, you know, the generative nucleus is going to divide and then make 2 cells. And you call those 2 cells as the male nuclei. The tube nucleus does not do anything. It just simply die out, right? So if you go to the next stage, you can see it clearly here. You can see it very clearly here. Number 17, that is 2 male nuclei. 2 male nuclei, and then number 18 is tube nucleus. That's tube nucleus, 18 tube nucleus, right? 18 tube nucleus. So that's all for the male part. So moving to the female part now, gynoecium. Starting from gynoecium, we come here. Number 19. Number 19, 20, 21. What's number 19? Who's going to tell that? This is called stigma. Stigma. Shall we take blue color for that? Shall we take blue color for the female? Female is blue. Blue female. Ne? So stigma. Stigma. 20 is style. 21 is ovary. Wall of ovary. Wall of ovary. Wall of ovary. Right? Wall of ovary. Then people, something special happened here. There is a red color cell present. What is this cell, people? We can label it as the megaspore. Megaspore. Megaspore mom cells, ne? Mother. Megaspore mother cell. Megaspore mom cell. What is the ploid level, people? 2n. So what happened to that? Undergo meiosis. It undergo meiosis. Then forms 23. 23. 4 of 23. What are those 4? Who's going to tell me? 23. What they are? Come on. Come on, come on, come on, come on. Huh? Macho megaspore. Megaspore. I'll take that megaspore. Right? 23. Let me take 23 as megaspore. Or else you can say macho megaspore. You're not wrong. You are correct. Absolutely correct. Right? I'm taking 23. Oops. Megaspore. Megaspore. One word, I'll write it like this. Megaspore. Haploid, right, people? Megaspore is haploid. Then what happens? The 3 megaspores are going to form. They are going to die. These 3 are going to die. One remain. So the mature one remain. So it undergo mitosis. Mitosis. How many times it undergo mitosis? How many times it undergo mitosis? Once. Twice mitosis. Twice mitosis. And 3 times mitosis. 3 times mitosis. Mitosis happened 3 times. And then how many nuclei it forms, people? It forms 8 nuclei. It forms 8 nuclei. These 8 nuclei rearrange. They rearrange and then form, people, the female gametophyte. 27 is female gametophyte. 27 is female gametophyte. So what do you call female gametophyte? What do you call female gametophyte? What do you call female? What do you call that? No, not synergid. No. Very good. Not archegonia. Not archegonia. Embryo, no. Embryo, no. It is the embryo bag, neither. Embryo bag. What do you call that? Embryo sac. So this is embryo sac. Embryo. Embryo sac. Embryo sac. That's embryo sac. Now, people, we have some other cells surrounding here. So you have to find the names of them. It's like a lot of girls, ne? Lot of girls. So you have to find these girls, their names. Boys, do you know the names of these girls here? Girl 25, girl 26, girl 28, and girl 29. Huh? What do you call them? 25 girl. 25 girl, you can call as 3 synergids. Sorry, I'm wrong. 3 antipodal cells. Wrong, sorry. 3 antipodal cells. 3 antipodal cells. 26 central central cell with two polar nuclei. Two polar nuclei. 29 is synergid. 28 is that beautiful queen. What's that? Egg. Haploid egg is there. That's the egg. Beautiful egg. Right, people? So, this one, right? It's actually present inside, ne? It's actually present inside this whole thing. This whole thing is here. It's there. So now we have to see what happens. What happens? Two male nuclei enter. Two male nuclei along the tube. Along the tube, two male nuclei enter. So one of the male nucleus go with, one of the male nucleus goes with the two polar nuclei. The other male nucleus goes with the egg cell, So what happened here? That is a double fertilization. What do you call? Double fertilization. Double fertilization. Fertilization happened two times. Double fertilization. Right? Double fertilization. So what happened in the double fertilization? Male nucleus plus two polar nuclei. Two polar nuclei. Male nucleus go with two polar nuclei. Result is what, people? What's the result? Result is number 34. What is 34? 34 is endosperm. Endosperm. And then, people, when the next event occurs, what is that? Male nucleus plus the egg. Male nucleus plus the egg. What that forms? That forms this one. That is number 36. What is number 36? Embryo. Oh, zygote, ne? Zygote, ne, people? Zygote, ne? We'll take it as zygote. Right? That is zygote. What is the ploidy level of the zygote? 2n. Ploidy level of the zygote is 2n. Ploidy level of zygote is 2n. Now, this whole structure, you can call as the fruit. This whole structure is the fruit. Inside the fruit, what you see, people, 33 is the seed. 33 is seed. 32 is fruit coat. Fruit coat, neither. Fruit coat. So that's the life cycle. That is life cycle. So this is going to start all over again. This starts all over again. So let me show you the whole life cycle now. Right? Beautiful life cycle. Anthophyta life cycle. Right? Anthophyta life cycle. Hope right, this is good enough for you to revise what you learned earlier, right? So you got to be in touch. My full aim is to help you to revise these early theory parts so that we can do continuously papers, right? Not the last moment papers, but papers throughout the time period, right? Not last moment papers, because a lot last moment papers are not going to help you to go to a good score, and it's just going to stress you, right? Think neither you try to do everything at once at the last moment, right? So let's get ready little by little, step by step, right? You have to learn. So plant life cycles, life cycle of flowering plants. If you quickly go through the note here, let's go on highlighting the important facts. Let's highlight these important things, people. Um, plant life cycle. Oh, well, let's quickly go through that. Life cycle of flowering plants. Sporophyte is a dominant stage. Gametophytes are short-lived and microscopic. Sporophytes produce reproductive structures called flowers. The flower is specialized. Flower is a specialized structure with 4 whorls. This word is important. Whorl. Who's going to tell me the name of, who's going to tell me the meaning of the word whorl? What's called whorls present? What's the meaning of it? Can someone tell me what whorls are? Can someone remember what whorls are? Flower is a specialized shoot with 4 whorls. 4 whorls. What that mean? Huh? Whorl? Come on, you know that, ne? You know it. Whorl nani? Are a fish they have whorl, ne? Maluangi whorl kelakene fins, ne? This is whorls. Whorls means rings, right? Rings of leaves, modified leaves are here. Whorls, modified leaves, named sepals, petals, stamens, and carpels. Right? Whorls of leaves present. Right? And then flower is, sepals are usually green in color, and then protects the flower before it open. So the job of the sepal is to give protection. Petals are brightly colored in most flowers and aid in attracting pollinators. So wind pollinated flowers are not that greatly colored. And then petals are brightly colored. These sepals and petals are sterile floral organs. Stamens are the microphyles. Stamens. The stamens consist of stalk called filament and 2 terminal lobes called antharineda. Then the flower structure, stigma, style, ovary, carpel. Okay, Neelamai. No big issue here. Receptacle. Look at this. They have marked it, ne? The receptacle, right? Then anther is made up of microsporangia containing diploid microspores. Microspores develop into haploid male gametophyte by mitotic division within the anther, so that the haploid male gametophyte is the germinated pollen. All right, that's germinated pollen, people. Right, germinated pollen is the male gametophyte. Germinated pollen is the male gametophyte. Right? So it has 2 cells, tube cell and the generative cell. Right? We learn all these things. Right? Tube cell and the generative cell. Male gametophyte with about 2 cells and spore wall constitute a pollen grain. Carpels are the megasporophylls. Carpels are megasporophylls. And then carpels are megasporophylls. And then at the tip of the carpel, sticky stigma present. Swollen base of the carpel form the ovary. Swollen base is forming the ovary. Right? And then ovule produce 4 megaspores by meiosis. Functional megaspore develop into female gametophyte called embryo sac. Highly reduced microscopic structure. What is highly reduced? The female gametophyte is highly reduced. What do you call the embryo sac? The embryo sac. That is the embryo sac. The mature embryo sac consists of 8 nuclei contained within 7 cells, 3 antipodal cells present, 2 polar nuclei, central cell, 2 synergids, haridalamai. Transfer of pollen grain into mature stigma of the same species is known as pollination. So pollination, we have 2 definitions for seed plants and angiosperms. In some plant species, pollen grains are transferred from the anther of the flower to the stigma of the same flower. Self-pollination, right? What is self-pollination and what is cross-pollination? Self is what, people? Self means what? Within same plant, same flower, ne? Within same flower is self, right? And then the cross-pollination, what is? Pollination may be, pollen may be transferred from stigma to flower of another plant of the same species. Cross-pollination. Then self within the same plant, within the same flower or another flower of the same plant. Okay. Within the same plant, same flower or another flower of the same plant. Self-pollination. Right. So cross is like between different plants. Most angiosperm plants are adapted for cross-pollination. Right. And then significance of cross-pollination are these things. Look at these people. Some modifications are present, people, which promote the cross-pollination. What are these modifications? Heterostyly, self-infertility, unisexuality. What is called as heterostyly? Means, what is the meaning of the word heterostyly? Heterostyly, what are those 3 words? Heterostyly, self-infertility, unisexuality. You know unisexuality, ne? I don't have to describe that. Unisexuality, you know. Unisexual. Unisexuality. And you know also self-infertility. That means when the pollen fall on the same flower, they don't pollinate. But the question is, people, what is heterostyly? Right? What is heterostyly? Heterostyly is this. This is a flower. Let's take this as a flower. This is the gynosium, short stigma, anther long. That means the filament is long. Stigma short. Heterostyly. It can be other way around. It can be other way around. What is that? This is tall. This is short. So in either way, this discourages the self-pollination process. Because when the pollen are produced at a top level, people, rather than falling down, they blown away with wind. So they can go to a different plant, different flower. Same like that. When the pollen is at the lower level, there is no chance that it fertilizes the stigma of the same flower. So this promotes the cross-pollination. So heterostyly, self-infertility, unisexuality, right? You know them, right? Now you know them. And then significance of cross-pollination. What is the importance of cross-pollination? This is resulting, people, genetic variation, right? I told you, right? Genetic variation. So it can result people shuffling. Cross-fertilization allows shuffling of genes within a species, producing new genetic combination, resulting more variation. That's like better for plants. Good for you also, Good for you, good for plants, right? So you have to find someone far away, not in the same family, not in the close by relatives, far away. That's always better. Right? Fertilization. Fertilization. So what are your plans for that? Do you have any plans for that? To find someone far away, ne? Not in the class, not in the next door, ne? Not in the same city, ne? Far away. Go far. Go to England. Ne? Go to England and find someone. Go to Russia, ne? And find someone. That's always better, ne? You will see it results super variations. Right? Mix up, blend, ne? It's like ice cream and dry fish, ne? New blend from America. Yes. Right. So it's like kind of a new trend now, ne? Most of the people go to Japan, Korea, and then settle down there, ne? They go to different countries and settle down rather than settling down here. They can get easily visa and all, ne? Citizenship and all, ne? That's kind of better way, ne? Right? Yeah, someone says from Maslow. Yeah, yeah, better, ne? You find an alien. Yes, that's a good plan. Okay. Right, my dear. So, fertilization. Now, this fertilization process, cross-fertilization, cross-pollination, cross-fertilization, right? So, fertilization process, let's look at that fertilization event here. Fertilization events, nucleus of the generative cell, the nucleus of the generative cell divide, forming two sperms, and then one sperm fuses with the egg cell, forming diploid zygote, and then the other sperm fuses with the 2n polar nucleus. This type of fertilization is called double fertilization. Double fertilization. After double fertilization, zygote develops, right? Zygote is going to form, and then it becomes the embryo. Significance of double fertilization is synchronizes the endosperm development with the development of the embryo. So both happen at the same time. Synchronization means what? Two events happen at the same time, right? Endosperm formation and the zygote formation is now happening at the same time. That's good. Why that is good? Because in cetus, whether the zygote forms or not, right? The endosperm is going to form in cetus. So it's an unnecessary wasting of food, right? So this can be avoided. Right? So that's a good plan, That's a good plan of the flowering plants, okay? And then the seed consists of embryo, endosperm with stored food. Seed are enclosed within the fruit. Fruit is an enlarged and developed ovary, usually after being stimulated by fertilization. In flower, if a flower has not pollinated, fruits does not develop. Okay, so these things we discuss. Right? Some more parts are there, people, regarding this parthenocarpy and parthenogenesis. Parthenocarpy and parthenogenesis. Right? Kapang. Kila kiwe mukadda. Yeah, what is kapang kiwa lamai? Yes, kapang kila kiwe. Fruits neda. If the fruits develop without fertilization is kapi. Right? Grapes, oranges and all. Right? Can be artificially induced. Banana. Right? Is parthenocarpic fruits. Then parthenogenesis is the formation of seeds without the fertilization process. Right? There you learn, people, something special. The what you said there, we said there in parthenogenesis. Look at this. In parthenogenesis, it can happen in three ways. Egg is resulted by mitosis. Egg is formed by mitosis, so that the egg is diploid. Or else haploid ovum fuses with the polar nucleus. ovum and the egg fuses. Third method, genetic content of the egg can be duplicated. N egg become 2N. It make 2 nuclei and those 2 nuclei fuses together. So in somehow it can develop into a seed, right? Without a fertilization process, that is the parthenogenesis. Significance of development of seeds and the fruits. Okay, I think people, the importance of that fruits, right? So read that part, read that part. So next week, I think we can go for a different section in the plant. You know, from paper 15 onwards, we are starting animal lesson also for this discussion. So then we are starting now coordination in coming Wednesday, right? Week after we start coordination lesson, right? I think then we are not behind. I check with the other classes; they also do the same level. So with couple of extra classes, I think we can cover up most of the content in the coordination lesson, right? So I think we can finish at the right time before August, right? So it's very important to do the papers now itself, right? Don't try to wait until the last moment. This is not going to be like 25 batch, right? The exam is going to be held on August as they have announced. So we have a short time period, so we have to get ready, right? So don't cut this session, right? I'm telling you, if you do this, I'm guaranteeing your result. Guaranteeing your result. Haridhar, if you cut this, no guarantees, right? You won't be able to go for a good result, right? And this is the only class, only class that you have the weekend session, and we do a lot of questions there, and then people extra classes, and then also the night sessions, right? You don't get this attention from any other master, so you stay and then complete all the components, right? This is really important, right? So shall we move to the part fungal life cycles? Fungal life cycles. Let's go for that. Fungi. Fungi. Now, people, fungi, life cycles of fungi. Fungal life cycles. Fungi. Fungi, ne? Fungi or fungi. How do you like to call? Fungi or fungi? Right? People, fungal life cycles. You learn this. Fungal life cycles, life cycle details you learn. Let's see what you learn there for fungi. Characteristic features of fungi. What are characteristic features of fungi? Prokaryotic or eukaryotic? Can someone tell me whether fungi are prokaryotic or eukaryotic? Come on. That's like a stupid question to ask from you all. I know you know the answer. Prokaryotic or eukaryotic, Lamai? Fungi? You know that, you can see it clearly. They are eukaryotes. Now, fungi are heterotrophic. We say that people, they are absorptive heterotrophs. Fungi are absorptive heterotrophs. Why do we call fungi as absorptive heterotrophs? Why do we call fungi as absorptive heterotrophs? Right? Why do we call them as absorptive heterotrophs? Now, whenever an organism is having a heterotrophism, it has to absorb, it has to absorb, it has to take nutrients from outside. It has to take nutrients from outside, right? When it take nutrients from outside, when it, sir, will the sperm have flagella? No, neither. Fungal, I mean, plant life cycle, flagella, no flagella, no flagella, no flagella. Not even, I mean, they are just nuclei. So how can be the flagella when only nucleus is available? No cell membrane also. So, okay, someone asked a question. Okay, Right. Now people, fungi, they are heterotrophic. Now what do you call as heterotrophism? They have to take the nutrients produced by the other organisms. So they take nutrients in. So when they take nutrients in, right, they have a cell wall as a barrier. So all these organisms, heterotrophic organisms who are having a cell wall like this, heterotrophic organisms who are having a cell wall, they can't carry out phagocytosis. Now think of amoeba. Think of amoeba, right? Think of amoeba. It's heterotrophic too, but it doesn't have a cell wall. So when it doesn't have a cell wall, it can take these outside materials, it can take these outside materials into the cell. It can take outside materials into the cell, right? By this phagocytosis. By phagocytosis. All right? By phagocytosis. But this guy can't do it. Right? This sort of a large particle, when it is available, it can't take it in unless it secretes enzymes and digest it and absorb the simple nutrients and absorbs the simple nutrients. So this kind of nutrition where they digest the particles outside and absorb the simple nutrients into the cell, we call it as the absorptive nutrition. And that is because of their cell wall. You understand that when they have a cell wall is working as an obstacle for carrying out phagocytosis, right? So not only fungi, but also we said that some bacteria with a cell wall, they also show, heterotrophic bacteria with a cell wall also shows absorptive nutrition, right? So it's not only limited to fungi, but you have to keep in mind, I don't say that all bacteria shows absorptive nutrition, because some bacteria are there, they are photosynthetic. Photosynthetic bacteria are present. Now, if you take cyanobacteria now, cyanobacteria is photosynthetic. Cyanobacteria is photosynthetic. So they don't have to show absorptive nutrition. So the rule is heterotrophic ones with a cell wall. Harani? Right? Clear, ne? Okay, look at these people. So eukaryotic. Cell walls are made up of chitin. They are absorptive heterotrophs, cellular enzymes which aid in breakdown of complex molecules into small molecules. Different types of different species live as decomposer, parasite or mutualism. Few are unicellular, hyphae present, and then septae. Look at these people. Look at this. The cross walls present neither. What do you call the cross wall? Singular term is septum. Cross wall, the term is septum. So you can see that some are sinusotic. Sinusotic means what? Sinusotic means they don't have a cross wall. No cross walls, right? Sinusotic. Actually, when cross walls are not present, when cross walls are absent, there is no boundary for the cell. No boundary for the cell, right? So therefore, it's kind of people multinucleate. Many nuclei present because there is no boundary for the cell. But on the other hand, people, when it becomes septate, you can see clearly a wall demarcates the boundary. So wall is there, right? And then people in fungi, some fungi are special because these fungi are having pores in the cross wall. The pores allow the flow of nucleus and other materials through from one cell to another, This is like an amazing feature of fungi. It's an amazing feature of fungi. You know why it's an amazing feature? Because fungi can grow people very fast because of this. Fungi can transport all their nutrients to one point. I taught you earlier, about the mama fairies rings. Remember fairies rings? Fairies like the fungi grow in the arena where the fairies come and dance. Why that happens so? Because when the fungal filaments, when they grow outward, the central filament die. The fungal filamental network encroaches the new area, outer area with fresh resources like this. Whereas the central part of this mycelium die, central part die. Right? So what happened, people? When mushroom comes like this in a ring. Mushroom comes in a ring like this. So what they call the fairies ring, fairies rings. Now, when a mushroom it grows quickly within couple of hours. It is because the cross walls and the pores present in the cross walls are allowing the transport of nutrients from the entire network to one particular growth point, so that the fungi, they have an unusual growth rate. Right? So we learn those things. Right? Septum has a hole which enables the movement of mitochondria, ribosomes, and nuclei, etc. Fungi lack septa known as sinusotic. Fungal hyphae produce mycelia. Some fungi produce hostoria. What are hostoria, people? Look at this. Hostoria are absorptive structures. When they grow, they can absorb nutrients from the plant cells. Right? Sucking part is like a straw, Right? It's like some straws. Suctorial parts, sucking parts. Right? They suck nutrients from the plant cells, like you drink with a straw, some lemon juice. These hostoria sucks from the plant cells the nutrients, right? So hostoria present. Ooh. Right, people. It's time to learn about this Chytridiomycota. Right? You learn a lot of mycota, Chytridiomycota, Chytridiomycota life cycle. Let me see whether we can, Chytridiomycota life cycle. You don't have to learn the life cycle. What you need to know is the general features of fungi. Characteristic features you learn. Look at these Chytridiomycota features, people. Chytridiomycota features. Chytridiomycota. What do you say, people? Chytridium aquatic, right? Terrestrial, aquatic or terrestrial. Some are decomposers while others are parasites. Decomposers and parasites. What do you call decomposer? They grow on dead organic materials. Parasite means they are growing on live organisms. Neither multicellular, unicellular, when multicellular, sinusotic. Now you understand that. Sinusotic. They produce zoospores. Zoo means animal. Their spores are animal-like spores. Right? They are animal-like spores. What do you call these stuff? You call this as zoo spore. Zoospore. Right? Chytridiomycotes, they have zoospores. So let's look at this life cycle. Let's look at this life cycle. Right? So this one is, I'll use red color. This is the mycelium. We can call sporophytic mycelium. Sporophytic. Again, a 2N mycelium, okay? Sporophytic 2N mycelium. Sporophytic mycelium. You don't have to know this life cycle. I taught you. And then people, this sporophytic mycelium that forms zoospores. In one way, this undergo meiosis. On the other way, it undergo mitosis. Like, let's take that mitosis happen. Then it forms a zoospore. That zoospore can grow back into mycelium. In meiosis, if happen, It forms once again zoospore. That one is a haploid zoospore. This one is a 2N zoospore. So they can form two types of zoospores, haploid zoospores and diploid zoospores, right? Now, once haploid zoospores are produced, this forms a gametophytic mycelium. You don't need to know the life cycle. Gametophytic mycelium. But gametophytic mycelium, that's not the important part. Now, they make people male and female gametes. They make male and female gametes. They unite and then form the mycelium back again. You don't have to remember this life cycle, no need. I'm repeating, you don't have to remember the life cycle, but you have to understand, people, these points which they have mentioned here, right? They say that they are zoospores. So if you understand this word zoospore, that's my aim by teaching you the life cycle, right? For you to get familiar with this word zoospore, right? Which are flagellated, and it's for reproduction. It can be sexual reproduction or it can be asexual reproduction. Cell walls are made up of chitin, that's like a fungal feature. Ne? Some of them form colonies with hyphae, while others exist as single spherical cells. Lamai, that's like special feature. Ne? Some are colonial, some are colonies, some form colonies with hyphae, while others are single spherical cells. Right? That's all what you had to learn about it. That's all what you had to learn about it. Right? Now we can go for zygomycota. Zygomycota. Zygomycotes, right? Zygomycotes. Can you tell me one example of a zygomycote? Who's going to tell me one example of a zygomycote? This is nice, ne? Around my head. I'm like God now, ne? Oh, fungal God, right? Muca. One of you say muca, rhizopus. That's correct. So if you look at this life cycle, if you look at the life cycle of zygomycota, life cycle of zygomycota, life cycle of zygomycota, life cycle of zygomycota. Look at this. This is the mycelium. No septine, no septae, many nuclei present, no septine, no septae, oops, no septae, right? Multinucleated mycelium. And then from here, some vertical filaments grow and they swell and then transform into a sporangium. They transform into a sporangium like this. Now, the important thing is this endospore and exospore story. Endospore and exospore story. What do you say about the spores produced here, people? Students, spores that are formed here, they are formed within this structure. So what do you call those spores then? Endospores or exospores. Kavisha, what do you call those spores? Produce inside or outside? Kavisha. Mukarram. Mukarram. Fazil. Mukarram. I think I have pronounced your name correctly. Tahira. Fahmi. What do you call these spores? Endospores or exospores? What do you call them? Endospores. That is correct. Endospores. They are endospores, right? These endospores are asexual and they can grow here. So what do you say? Asexual endospores are produced by zygomycota, right? So that's the very simple asexual reproduction process. Sexual reproduction happens between genetically different Fungi. So these genetically different. So to show the genetic difference, people are using red color and black color, but you don't understand that there aren't any fungi, right, in black and red, right? I'm just showing you people the genetic difference with the color. They grow towards each other. These gametangia. Gametangia. They come and contact with one another. The gametangium unite, and then people exchange their nuclei. They exchange their nuclei. This process is now forming a new structure where the nuclei, both nuclei present inside, and they get united later. The nuclei are going to fuse. This we call as the zygosporangium. We call this as a zygosporangium. Right? This is a sturdy structure. Right? What they say, people, zygosporangium. Neither. This is the zygosporangium. Look at this. Look at this. Asexual reproduction produce sporangia, which is genetically identical haploid. Right? Sexual reproduction, a zygosporangium is produced, which is a sturdy structure. Sturdy means what, people? Tough structure. Produced by plasmogamy and karyogamy. Plasmogamy means cytoplasmic fusion. Karyogamy means nuclear fusion. Zygosporangium is resistant to unfavorable environmental condition. Zygosporangium is multinucleated structure which resist of drying and freezing. They are metabolically inactive in adverse environmental conditions, and zygosporangium produces genetically diverse haploid spores. Right? That's it. That's all. Right? That's all. Right? So this is zygosporangium. Later, right? Later it germinate, forming a new sporangium. And once again, people, like before, spores are going to form inside. These are sexual endospores. These sexual endospores are going to disperse. They're going to disperse and then form the mycelia back again. They're going to disperse. So here, both sexual and asexual spores are endospores. Both sexual and asexual spores are endospores. Right? Zygosporangium forms during the sexual reproduction. That is what you need to know. Right? And then, people, moving to the ascomycota. Ascomycota, check that. Marine and freshwater, parasitic-symbiotic, those things you have to like pay special attention, people. Parasitic or symbiotic, decomposers, unicellular, filamentous, or multicellular sexual reproduction conidia are produced. Sexual reproduction conidia. So let's pay attention for that. Sexual reproduction conidia. Sexual reproduction conidia. Conidia. What are conidia? Conidiospores. Look at this. Asexual conidia, not sexual. This is the filament, septate. What is the special feature? Po present in septate. Po present in septate. So nuclei can flow between cells. And then once again, people, this filament is also forming vertical filaments like this and build up the sporangia. Now the special feature is they form some flask-shaped structures, some tube-shaped structures, and they form, people, spores towards outside. That's like a special feature. Previously, the asexual spores are produced inside. Now the asexual spores are produced towards outside. So what do you call them, people? Exospores. These are conidia, neither. Conidiospore. Conidiospores. What do you call? Exospores. Conidiospores are exospores. Here there is a special term. Here there is a special term. This one. Conidiophore. Conidia. Right? That's a special term. Conidiophore kiyanne meka. This is conidiophore. Phore. Is the structure that bear the conidia. Right? This conidia, conidiospore, or else you can say conidia, conidiospores or conidia, same. Haridam. But then they are using a word conidiophore. Conidiophore is that stalk-like part. They say it's a specialized hype. What is conidiophore? Specialized hype. Specialized hype. Specialized hype. It's a specialized hype that bear the sporangium. What do you call that? Conidiophore. Conidia or conidiospores are like a different term. Anyway, they germinate and then forms, people, the mycelium back again. Sexual reproduction is different. Once again, to show the sexual reproduction, we are using a different color. We use a different color to show that they are genetically different. And here, people, we can see a gametangium is going to form, but this gametangium, you can identify male and the female. Male and the female can be identified. So male gametangium and the female gametangium present. Female gametangium is having a small filament-like part. So we have names. What do you call? Antheridium. Antheridium, this guy. Male one. What do we call the other one? Ascogonium. Ascogonium is the other one. Ascogonium, into the ascogoniums, the nuclei are going to move. These nuclei are going to move into the ascogonium. These nuclei are going to move into the ascogonium. Next, some special events occur in the ascogonium. Ascogonium stretches out like this. Ascogonium stretches, and inside the ascogonium you can see those paired nuclei present. And then ascogonium is surrounded by other fungal filaments. Right? It's like ascogonium is now pregnant, right? Protected by the others. Protected by the others. Right? So what it forms now? It forms something called as the ascocarp. What do you call this whole structure, people? Ascocarp. Ascocarp, this whole structure. And this one is a dikaryotic filament. It's a dikaryotic filament. This dikaryotic filament now forms what? Asine. This form, people, spores, sporangia. It forms sporangia, right? This filament forms sporangia like this. Then we call these sporangia as ascus. Ascus. You can see 8 spores are going to form within this ascus, and these spores are going to disperse. These spores are going to disperse, and they're going to grow back into the filament. You don't have to learn the life cycles here. Life cycle details are not necessary, but if you can understand the basic things, that's good enough, right? What you have to do is you have to understand this life cycle here in sexual reproduction, fusion of sexually differentiated gametangia. Look at that, sexually differentiated gametangia take place. Sexually differentiated gametangia. Ascospores are produced within asci. Generally, there are 8 ascospores, and most of these fungi produce ascocarp. Ascocarp, I mentioned it. Ascocarp, people. Ascocarp I mentioned here, right? So they form ascocarps. So we move now to the last life cycle of fungi. What is that? That is the Basidiomycota life cycle. Basidiomycota life cycle, right? Let's check Basidiomycotes. What do you know Basidiomycota? They say, people, asexual reproduction is not a common event in most of the Basidiomycota. They have, but the example that we discussed, the agaricus, asexual reproduction is not common. Sexual reproduction only, right? Sexual reproduction process happens. That's also a bit unusual type of sexual reproduction. Look into these details a little bit, people. Characteristic features. Oops. Characteristic features. Look at this. Characteristic features. They are terrestrial, major decomposers, and some are symbionts. And then filaments with septae and dikaryotic. Dikaryotic. Dikaryotic mycelium is the dominant stage. They produce fruiting bodies called basidiocarps. Produce basidia on the gills. Produce exogenous basidiospores. Exogenous basidiospores. Right, look at this. This is one filament. This is one filament, ne? And this is another filament. Somehow these two filaments with nuclei, they come in touch with one another. They come in contact with one another. So they exchange nuclei. So when they cross each other, they form a cell. From the contact point, they form a cell. That's a new cell. This is going to form a new cell, 2N cell. This region forms now a 2N cell. This cell is going to grow. And this one is now forming a new dikaryotic mycelium. Where are those nuclei present inside? Where are those two types of nuclei present inside? Like this. This filament, right, grows into a mushroom. It grows into a mushroom now. It grows into a mushroom. And on the underside of the mushroom, you can see the gills available. On the underside of the mushroom, what is available? Gills. We call them as gills. Right? Gills present. You take one of the gill and you enlarge the gill. You can see, people, there are some special structures. We call them as the basidiocarps. What do you call? Basidiospores. This whole thing is the basidiocarp. Right? Wait, this whole thing, what do you call? The basidiocarp. Basidiocarp. Here, people, this is basidia. Here, this one is the basidium. D-I-U-M, singular. Basidium. Basidia, plural. Harida. And here you can see, people, those two nuclei present. They unite. So, and they undergo, people, meiosis. They fuse and undergo meiosis. And by meiosis, they form 4 nuclei. They form 4 nuclei. And these 4 nuclei, they arrange into spores. They arrange into spores by growing outward like this. So we call them as exospores. They form 4 spores outside like this. Right? What do you call them, people? Exospores. You call them as exospores. These exospores, they are basidiospores. Exospore. These are exospores. what do you call them? Basidiospores. They grow and form new filaments. They grow and form new filaments, right? So that's about the life cycle. That's about the life cycle of Ascomycota, right? That's the life cycle of Ascomycota, right? So people, in this paper number 13, it has the life cycle details, plant life cycle details, fungi and their details. Another important part is the cell cycle. Another important part is cell cycle, right? I think people, cell cycle you can study, ne? Can you study now? Couple of times we skip this cell cycle. Previous time also I skipped cell cycle, ne? We'll take it into the next day discussion, ne? Cell cycle, and also water potential. Some questions are there too. I want you to now start studying water potential, because our next discussion is going to be water potential, right? So we'll discuss this paper next week, right? Through that certain parts are there which we have not discussed, but this paper is slightly different. I gave it as an overall paper from the most of the parts that we discussed as a mix-up paper, right? But next paper onwards, we'll prepare it for certain target sections, right? We'll discuss what we are going to make the paper for the paper number 14. Paper number 14, we'll talk about the design of it next week once we discuss the parts, right? Then thank you very much for your participation and joining with this short discussion. Your suggestions are going to be really helpful. If you suggest certain sections to discuss, I can then give the priority for those parts, right? And then thank you once again for the joining with the discussion. And anything, people? Is there anything special that you want to say? No. If anything special is there, let me know. Right? You can WhatsApp. Yeah, you can talk with me. Tell me. Right? We'll take them to discussion. Thank you very much. Right? Have a nice time. I'll see you all soon. Bye.